Good morning students. Welcome to the next video class children. How are you all children? So I hope all are fine. Okay, good. Have you remembered that previous class portion children? Yes. Which are the topics we had completed children? Can you able to say? Yes, good. First one, learn that bacteria staining, isn't it? Staining procedure, the steps involved in staining procedure. Then how we are differentiating this gram-positive and gram-negative type of this bacteria, is it? Then next one, what is the differences between these two type of this bacteria, isn't it? Clear? Have you learned that question answer? Say yes or no children? Yes. Yes, good. Next one, we had completed that is the life processes in bacteria, isn't it? The life processes in bacteria. So there comes under in your syllabus based the respiration, very good, that's a nutrition and then reproduction, isn't it? The last class we had completed that too, isn't it mom? Very good. What's the two things are completed? Nutrition and respiration. What is the major two types of respiration in bacterial cell? So there is, very good, there is a anaerobic respiration and aerobic respiration. Clear? So there also we had completed that zone of the topic. Very good. So there is a, what do you mean by facultative anaerobes and what do you mean by, very good, obligate aerobes, isn't it? So these are the things. And then finally we had completed that zone of the 5-mark question. That's very important one of the question children. Nutrition, isn't it? The nutrition, what are the two major types of nutrition children? Very good. So autotrophs and heterotrophs, isn't it? Autotrophic mode of nutrition and another one is heterotrophic mode of nutrition. Autotrophic mode of nutrition also they comes under two types. Can you able to say? Yes. What are the two types? Photoautotrophic and hemoautotrophic. Understood ma'am? Yes. This photoautotrophic also they comes under two. What are they? Photolithotrophs and photoorganotrophs. Clear ma'am? So all should learn thoroughly. This is the previous class portion. This is very important one of the 5-mark question. Previous already I said the steps involving in, very good, that's a staining procedure of bacteria. That also one of the important 5-mark, and a difference between gram positive and gram negative type of bacteria. Clear? Yes. Then we are going to move. This is the next topic. So that's life processes in bacteria, reproductions of bacteria. Okay. So if we are looking, this is the reproduction in bacterial cell means. So here there is mainly they comes under two methods children. One is asexual and another one is a sexual method of reproduction. Okay. Asexual means so they not involving there is a gene transformation, the DNA transformation. Okay. The asexual method they mainly they comes under in the two types children. One is binary fission and another one is endospore formation. Okay. So this is the first type. So there is a very good binary fission and then second one is endospore formation. Clear children? So these are the two asexual method of reproduction takes place in bacterial organisms. Understood? Let us learn that one by one. Binary fission. I mean, so the bacterial cell during that is the unfavorable situation. Okay? So there is a unfavorable situation. So there they producing in that is the endospore formation. Favorable situation, so this is that asexual reproduction that takes place in a binary fission. So that's a favorable situation. So here they takes place in a binary fission. So how does it takes place? This is the binary fission means first, so this one, the nuclear material is dividing into two. Okay. Then followed by, so here the formation of a small constriction of that particular cell. Then after that, finally they dividing in a two daughter nuclei. That's a two daughter cells. Understood, huh? First nuclear material is dividing and second one followed by this is the cell. So here that small constriction takes place and formation of this constriction and followed by the separation of these two cells. Clear, ma? So this is we are called as the asexual method of binary fission in bacterial cell. Understood? So this is the diagram. From this diagram you should understand more. Next one, endospore formation. Already I said unfavorable situations, unfavorable conditions only they produces the bacterial cells or they produces in a which one? Endospore formation. What do you mean by endospore? Okay, so that means by the thick-walled resting spore is called as the name is known as endospore. Clear? What do you mean by endospore? The endospores are thick-walled, very good. A resting spore is called as the name is known as endospores. Clear, children? Say yes or no? This endospore producing, there is the bacterial cells or some of the examples they are given in your syllabus, children. The first one is Bacillus megaterium and Bacillus fasciaris and Clostridium tetani. So these are the some of the bacterial cells so that can producing in that asexual reproduction that takes place, that's a endospore formations. Understood, ba? Then after that, favorable situation, the favorable condition, so that can be germinate and form, that is the bacterial cell. Understood, ma? So this is the two type of this asexual method of reproduction in bacterial cell. Asexual mainly that's two types, children. One is binary fission and another one is endospore formation. How was it takes place in that asexual method of reproduction in bacteria? So we had to know this two types. Are you clear? Say yes or no, children? Yes. Next one we are moving in, that is the second major type of this reproduction in bacteria, that's the sexual reproduction. Okay. Here if we are making this is the sexual reproduction in bacterial cell means, so not formation of this fusion and formation of this gametes. Okay. Normally sexual reproduction means, so there is the formation and fusion of the gametes. Okay. But here this is the bacterial, that's a sexual reproduction. So without there is the very good the formation and fusion of the gamete cell, right? Only the genes are transferring. Okay, so that's a gene recombinations. So the genes are transferring from one bacterial cell to next one. Clear, children? So that's the method only they take. So through recombination method, the genes only they transferring through this sexual method of reproduction in bacteria. Is it clear, children? Say yes or no? This sexual method of reproduction, they comes under in three methods, children. First one is conjugation, transformation and transduction. Okay. What are the three methods of sexual reproduction in bacteria? Please try it, children. Conjugation, transformation and transduction. Understood? Yes. Let us say one by one. First method, conjugation. Okay. Conjugation, so this method that is discovered, that's a person name is known as Edinburgh, and then so that's another one more person, Edward, so that is the Tatum, L. Tatum. Okay. So these two persons only they proposed or discovered, that is the very good the conjugation method of sexual reproduction in bacterial cell. Clear? So in the year of 1946, okay, so that person which type of this bacterial species they are using means, so they are using in an E. coli bacteria. So that's mean by Escherichia coli. Okay, that bacterial cells only they can use for this method of the conjugation method of asexual reproduction. Understood, children? Yes. Then last class I told you, so that's that's a pillai. If we learned that is the ultrastructure of bacterial cell, then we learned that is the pillai. Pillai means additionally one special type of the pillai is there. That pillai name is known as sex pillai. Do you remember this concept? Say yes or no? Yes, very good. Clear? But here, this is the conjugation method of sexual reproduction in bacterial cell. Clear? So first one, here they are selecting in the same species of the bacterial cells. But first one, the conjugation method. So here, first one of the cell is donor cell and another one is the recipient cell. Okay, so this one is the donor cell and another one is the recipient cell. First one, donor cell, they has that is the F plus factor. F plus factor means, so they has that is the fertility factor. Okay, F plus factor, so this is the fertility factor is there. Okay, then the recipient cell, the absence of this F plus factor cell, right? There is a fertility factor is absent. Clear? Then first one, how this is the conjugation is going on. A conjugation method of gene transferring in that bacterial cell means the donor cell makes that contact of this recipient cell through that is the sex pillai. Understood, children? So this is the contact only. The donor cell make it in a contact of this recipient cell through that is a pillai. This pillai, so pillai means just one fiber-like nature. Okay, so this pillai finally they grows and forms that is the conjugation tube. Okay, so this is the one tube-like nature is there now. Two bacterial cell connecting, that's one tube. This is known as conjugation tube. Understood, children? Say yes or no? Yes. Next one, already I said, this donor cell they having in that plasmids of F plus fertility factor is there. Clear? So here this is the recipient cell without that's a fertility factor of this plasmid. Understood? How this is the DNA plasmids transferring from this donor cell to recipient cell. Through this is the conjugation tube. Clear? Through this is the connection of these two. This is the donor cell, this is the recipient cell. These two cells, the conjugation tubes only, the one strands of this DNA, the one strand of the gene, the DNA, transform from donor cell to recipient cell through the conjugation tube. Clear, children? Now, this is the one of the double-stranded fertility factor is there. One of the double-stranded plasmid. Okay? Then, donor cell, only one strand is give to this is the recipient cell. Then the next second strand is the recipient cell only synthesized that. Clear, children? Say yes or no? But here, that's the first one. Donor cell, they have that is the double-stranded plasmid. They have that's the F plus factor. That's a fertility factor is there. How the genes are transferring from donor cell to recipient cell? Through the conjugation tube. One strand of this is the two strand. Okay, the one strand is through the conjugation tube, they giving to this is the recipient cell. Then automatically recipient cell, they make it in another one more strand cells. Understood, ma? So this like only they going on that gene transfer through the methods of conjugation. Clear, children? Say yes or no? Now finally these two cells, so this is the donor cell and this is the recipient cell. Both the cells they have that's a F plus factor. That's a fertility factor is now be present in this both the cells. Donor cell as well as this is the recipient cell. Are you clear, children? Say yes or no? Very good. So this method of gene transformation, that's the first one is the very good. So that's the method name is known as conjugations type of sexual reproduction in bacterial cell. Next one we are going to learn about this is the second method, transformation. Okay, so that's the name is the second method is transformation. This method, that's one of the bacteriologist name that's discovered as the person name is known as Frederick Griffith. Okay, in the year of that's discovered that's in the year of 1928. So this person they using that's the organism. This is the experimental work they are using that's the organism maize. Okay. Then which type of the bacterial cell is used for this transformation of this sexual reproduction in bacteria? This is Diplococcus pneumoniae. Okay, the Diplococcus pneumoniae. So these are the some of the one word children. So you must prepare this on the concept. Okay. The next, how this is the gene transferring. This is the method of transformation. Okay. Yes. First one already I said they are selecting this maize. Clear? This mice, first one, this person, they selecting or they are choosing or they form that is a two type of the strains from this Diplococcus pneumoniae. Okay. First one is, that's a bacterial cell, they formed in that is the one of the S type of the strain and another one is, so that is the R type of the strain. Okay. So these is two strains. The first one is S type and another one is R type. I mean S type means that's a smooth cell. Okay, the S type of the cell is smooth cell, but that has the DNA is a virulent type. Understood, ma'am? Then R type of the cells means, okay, so that is a rough type of the cell, but that is the avirulent type. Clear? I mean, first they are choosing these two strains. I mean created these two strains, injected into the mouse, what's the responses coming? The first one, that person, that's the bacteriologist name, can you able to say? Yes, good. So that's the person name is Frederick Griffith. Okay, yes. That person, first that person is they selecting, they using this is the gene transfer. Or that's a DNA transfer from one bacterial cell to next one. First that person is they using the strain of a rough strain. The rough strain is injected into the mouse. The mouse is alive. Okay, so that mouse is survive. But this is the smooth strain means already I said the characteristic features of the smooth strain is the virulent in nature. The R type of the cells means, so that is the avirulent, non-virulent in nature. Clear, children? Then second time, the smooth type of the cells are injected into the mouse. At that time, the mouse is going to die. Clear? The mouse is died. Understood, ma'am? Then after that, the third strain, that person is the smooth strain heat killed. The heat killed process they using of this the S type of the strain. I mean this heat killing process means they destroying of this protein of this bacterial cell, but that's the DNA is active only. Understood, ma'am? But the S heat killed type means that's the denaturing of this protein of this bacterial cell, but that one DNA is active position only. Clear? But this type of this strain is injected into the mouse. The mouse is that is not going to die, so that is live. Clear, children? Then finally, how to transferring of this DNA material from one strains to next one. That's a step is here that person is they mixed two. This is the two item. One is the heat killed S type of the strain. And also there is the R type of the cells. What's the things going on in this region means? All of you listen carefully. The S type of this DNA is already I said protein only denatured, but that is a hereditary material. DNA material is active, isn't it? That virulent type of the DNA, the S strand, they have active, that's the DNA. That DNA is transformed to this is the which strand? Very good, R strand. Understood, children? So this is the heat-killed yeast type, that's a gene. They transformed to that's a R type, that's a rough type of the cell. Okay, children? Then what will happening means, so this type of this injected into this, the strands are injected into this mouse, so that can be dyed. Clear, children? So this is the way only the one gene is transferring from that is another one bacterial cell. Okay? What do you mean by transformation means the transferring of DNA from one bacterial cells to next bacterium, that's known as transformation. Clear, children? Say yes or no, ma? So this is one of the very important 5-mark questions, children. So all should learn the questions thoroughly. Clear? Yes. Then there is a third method of this sexual reproduction in bacterial cell. That's a transduction. Okay? Transduction. So this method discovered or proposed by Zinger and Liedenberg. Okay? So this is the two persons only. They discovered this is the method of the sexual reproductions in bacterial cell. In the year of 1952, they are using this is the bacterial cell is a very good. So that's a Salmonella typhimera. Okay, so this is the bacterial cells only. They are used for this is the method of this sexual reproduction in bacteria. Okay, next one we are going to learn about this is the third sexual method of bacteria. First one, what do you mean by transduction? Okay, listen, children. The phage mediator, this is the phage, clear? The phage mediated DNA transfer, that's known as transduction. Understood, children? What do you mean by transduction, children? The phage mediated DNA, that's known as very good transduction. Okay? So this transduction is, they comes to two types, children. One is generalized transduction and another one is specialized transduction, or another one names also restricted transduction. Clear, children? Will you say this the two type of this transduction? Very good. First one is generalized transduction and another one is very good specialized transduction. Specialized, another one names also, they are called very good. Tell me, restricted transduction. Okay. Let us see that's one by one. First one, generalized transduction. The ability of this bacteriophage, clear? The ability of this bacteriophage, the DNA material that is transformed into that, okay? The transforming into that is not they mentioning that position. So in any places they inserted into the bacterial chromosome, that is known as generalized transduction. Okay, not point out in that is the particular position, any regions, that is very good. So that is mediated in that DNA to this is the bacterial cell, that's known as generalized transduction. Clear, children? If any places that's a phage mediated, that's a DNA is transferring into the bacterial cell. The next one, specialized transduction means the ability of this bacteriophage, the DNA is transferring into this is a bacterial cell in that the specialized region or that particular position. Understood, ma? So that's known as specialized transduction or that is a restricted transduction. Clear? So these are the two types of this. So the transduction, so the first one is generalized transduction and another one is the specialized transduction. So detailed they are given, this is the diagram also. First one is the generalized transduction and then the next one is specialized transduction. Clear, pa? Yes, very good. So today's class, we need to prepare First one, what are the two common methods of reproduction in bacteria? First one is asexual and another one is the sexual method of reproduction. The asexual, so there is the formation of this binary fission, and another one method is the endospore formation. What do you mean by endospore? Very important, one of the 2 mark questions, children. So all should prepare thoroughly. Next one, very good. Sexual method of reproduction. This sexual method, 3 types of this. The 3 methods of sexual reproduction in bacteria: conjugation, transformation and transduction. Okay, conjugation is individually they will ask, that's one of the 5 mark question with the diagram. Next one, transformation. What is transformation? That's one of the very important 2 or 3 mark question. And then the second one also they will framing, that's one of the 5 mark question along with that diagram. Then finally transduction, that's very important, one of the 2 mark question. What do you mean by transduction? Then explaining its types. So sometimes they will ask, that's a 5 mark pattern. Clear, children? So these are the questions, you must prepare it, and in between some of the 1 word questions are there, children. Please all of you read line by line. Okay, clear, children? Yes. The next I am going to give on that homework, children. Already I asked you to draw some diagrams in your classwork note, children. I hope all are completed that diagram in your classwork note. So today also I am going to give some diagrams. First one, so you have to draw, this is that asexual method of reproduction. These 2 methods, children. One is binary fission and another one is endospore formation. And the next one, each page you make it to draw. This is the 3 methods of sexual reproduction in bacteria. Clear? First one is conjugation method. Second one is, very good, it's a method of transformation. Then third one is transduction. Okay, each pages you should draw one diagram. The 3 pages can accommodate. Clear? Are you understood, children? Yes, very good. Thank you, students. Thank you very much.